Hi, and welcome to this episode 2 of season 2 of the rEView podcast. I'm Natalie Turner, and today I have Farouq Sharifpour here with me. Farouq, why don't you go ahead and introduce yourself and give the listeners a little bit of background as to who you are and what you currently are researching.
Hi, thanks for having me. My name is Farouq Sharifpour. I am a researcher in extracellular vesicles and molecular biology. I have also a veterinary background. So I'm coming from actually veterinary school back in 2008 when I started veterinary school and graduated 2014 back in my home country, Iran. And I did a fair bit of veterinary practice back in my hometown, and then I received a scholarship from the University of Adelaide to pursue my PhD. So it was when that I focused on molecular biology and synthetic proteins. My PhD was about developing molecular system to synthetically regulate the transcription in Toxoplasma gondii. And you know, Toxoplasma gondii is a zoonotic pathogen that can infect humans and animals. And when my PhD was finished, then it was when that I changed my focus to extracellular vesicles. And since probably 2022, I've been working on extracellular vesicles to uh, 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 almost solely, uh, and and when I started, I soon started to like this field of work. It's very open. There's a lot of possibilities in there. There's a lot of things that are still unknown that we could learn. And yeah, it was all probably by luck because, I never chose to, go any of this. It just happened right. I was on the probably on the path very randomly. So neither molecular biology nor extracellular vesicles. But I realized after that, I realized that, yeah, it actually it's a good combination to have, especially for future work. So
Yeah,
yeah.
absolutely. So, I mean, you mentioned, so you had this veterinary background, you were studying in Adelaide, you finished your PhD, and then all of a sudden extracellular vesicles happened upon
Yeah,
you. So what's the how and why behind that? Because you're now currently at James Cook University, is that correct?
I am.
Yes, and
That's
that
very
is a
moment.
long way from Adelaide for anyone who does not know Australia. The kind of
Yeah.
opposite ends of the country. JCU is in like far north Queensland. Or like, what is far north Queensland? It's Townsville, is that correct?
It's Cairns.
Or Cairns. I was
Oh
wrong, and I'm
yeah.
from Australia, so I'm
Uh,
sorry. That's
no,
JCU people.
you're right. Real right. Townsville is also far north Queensland, but
Yes.
it's JCU's problem because JCU has a lot of campuses. So
Yeah.
it has campuses in various cities, mostly in far north Queensland. Also, it also has a campus in Singapore. So,
Ah,
yeah.
did not
So,
know. Okay.
yeah. So I am in Cairns at the moment, but I
Okay.
probably soon move down to Townsville.
Okay.
To just had a bit of change of career, but I still will be in research. The thing is, yeah, it was a long way. You're right, from Adelaide to, I mean, it's such a long way from my home country to Cairns. It
Yeah.
was a long
Yeah.
journey. It all goes back to veterinary medicine and parasitology. So now I'm thinking about it. There are two missing links in my story. So the reason why I got into the I did my PhD in veterinary school at University of Adelaide. So veterinary background actually introduced me to that veterinary school at Uni Adelaide, right? And there I started on parasitology. Toxoplasma gondii is a apicomplex and it's a single cell parasite. Then I wanted to go to a parasitology lab to continue on parasitology. So Alex Lucas Group is a famous parasitologist in Australia and worldwide. So that's why you know, went to Lucas Group in here at JCU, and then they wanted to know about more about extracellular vesicle coming from helminth. So yeah, there was some missing links. So veterinary medicine, my connection with veterinary background and Uni Adelaide, and then parasitology from Uni Adelaide to James Cook University. So it so happens that Alex Lucas had this opportunity to work on extracellular vesicles from helminth, so that where I started.
Yeah, and I mean, kind of, I mean, helminths aren't exactly what you would think of in like the veterinary world, unless you're worried about them infecting or the host. So it's like you start working with these big organisms, I'm sure, in veterinary in
I
the
know.
veterinary world. I myself was working with plasma, extracellular vesicles from plasma from cows, very large animals,
I
veterinary
know,
approach,
yeah.
agriculture, and then you go to the other extreme where you're working with helminths. So they're tiny, they're worms, right? So
They are. I mean, some of them are tiny, some of them are actually pretty long and huge, for example, ascaris can, uh, especially we have ascaris in all species, like in mammalian species, right? Ascaris can be really long, and for extracting the extracellular vesicles out of them, you have to,
mm,
fit all these long, probably 1 meter or more worm into petri into flasks, into T175 flags, and then let them be for a while in culture medium to extract the EVs from culture medium from there.
so you brew the worms to get the EVs.
Sort of,
So
sort of.
they just secrete them. Is this part of their natural process?
Exactly, exactly. Worms can cause inflammation, right? Every pathogen can cause inflammation in their host at some point. But evolution wise, so they adapted a system or adapted a strategy to suppress this inflammation. So for them to be living freely and without hassle, they have to come with a mechanism to suppress the inflammation. So in that way, they secrete a lot of stuff. They secrete small compounds, and they secrete microRNA, they secrete proteins, and they secrete extracellular vesicles to do that. To just, it's a interspecies host pathogen interaction. So pathogen secretes some stuff, either proteins, small compounds, or EVs, so that they can taken up by host, especially in this scenario, intestinal cells, and then they trigger some pathways to suppress immune response, so that the immune response that's supposed to happen against this pathogen would not happen. So and they could live longer in our bodies or
Yes,
in the
so they
host
can
body.
survive.
Yeah, this
How
is very clever.
lovely.
Yeah,
Yeah, it's
clever.
clever and really a bit gross to think about what they are
Exactly. Yeah.
doing. But no, I know of research being done at JCU in the past that was all about the worms, how they infect the host, and potentially even as like a treatment for inflammatory bowel disease or related
Exactly.
things because they have that anti-inflammatory profile, whatever they secrete. So it seems that EVs have a role in this, obviously.
Mm. It does. It does. So nobody, especially IBV. So IBV is inflammatory bowel, sorry, IBD, inflammatory bowel disease. So almost no one knows how it happens. So it's idiopathic. Although I should correct myself because a recent publication, probably a few months back, they found some genetic mutation to cause, but it's just one factor. There's a lot of factors for that disorder to happen. Uh, but anyway, what it does, IBD to intestines, is very nasty and chronic inflammation that sometimes causes ulcer as well in the intestines or in alimentary tract. So at some points, it can even happen in esophagus. It's far from intestines. So, but the main similarity between all IBD forms is there's many IBD forms, so is inflammation and some pathways that trigger that inflammation. So researchers thought about maybe we can, interrupt that pathway, inflammation pathway, and we can, just treat the IBD in a symptomatic way, right? Not We can't probably treat the cause of the IBD anytime soon, but we can suppress the symptoms, right, by interrupting the inflammation pathway. So there is a lot of proteins known, chemicals that can trigger that pathway interruption against inflammation. One of the things that Alex Lucas Group does is leveraging that natural capacity of intestinal worms, right? And especially hookworms. So hookworms are some worms that actually latch onto the intestine and then kind of feed from the blood. So uh, so those worms produce proteins and extracellular vesicles that can carry some microRNA, right? Uh, and they found specifically some proteins that can have potentially anti-inflammatory properties against IBD, they're going to use it against a variety of chronic diseases, not only IBD but also diabetes and even arthritis. Yeah, this is the whole story. So you have to interrupt the inflammation, but with what? So with chemicals, we can do that with medicines. But Lucas' group approach is with naturally occurring proteins that coming out from worms, because these are natural, right? They know what to do evolution wise. So they evolved to produce some natural thing with the minimum side effect. They just uh, uh, adjust or tune the inflammatory response against themselves. So we're going to leverage that. We're going to hijack those proteins from them and produce them in the lab with different vehicles, and then probably introduce them as new therapeutics for IBD.
So there must be some kind of two-way communication going on for like there must be a feedback between worm and host and the environment that they're in, because I find it fascinating. There's got to be like a kind of homeostasis for the worm, so it's happy and it's secreting just enough of the correct molecules to make its environment safe for itself, and so it can continue to live and feed and.
I guess so. So, I mean, uh, I could come up with an analogy. So, you probably we live in tropics here, right? And it's very humid. It's very hot. At some points, it's
I
unbearable.
know. Yeah.
Yeah, you know that. So, when you think like living in an environment, you have to either adapt yourself or you're going to find a shelter, to provide you your standard conditions to live. So we live in homes, we use AC, we use dehumidifiers to, adjust the environment to ourselves. And that's exactly what the worms do, in our body, or any kind of parasite. So they adjust their small environment, their kind of, we can say, their bubble of life, but they bubble around them to, avoid those harsh conditions that host imposed on them. Okay, to some connect. There may be a two-way communication, there may be, but I think my feeling is no, the wormers are actually hiding themselves from and the best they can do that, they can survive longer. That's how it goes. And it happens also in even intracellular parasites. For example, in Toxoplasma, you know, they have a good way of living inside their host without ever making any symptoms, without even the human body knows that they are there. They live in kind of dormancy, and they did you know that even 1 in 3 people in the entire world is estimated to have Toxoplasma, but it's most of them are latent. Yes, most of them are
Wow.
latent. Yeah, exactly. So, but they can cover themselves, they can hide themselves so that the body cannot recognize them. Otherwise, the body would attack them, and attack them, and they wouldn't let them live freely. So that's what parasites actually do the best. So they cannot regulate their uh, you know, microenvironment inside a big environment like us. They try to regulate their microenvironment to be hidden against the inflammatory response of their host, and also to, mitigate the harsh conditions that they may be opposed to.
And I mean, this obviously we're talking about the worms quite a lot. Just in case our listeners were not aware, you've just had 2 papers published very recently, within, I guess, the last few weeks or months. One in the Journal of Extracellular Vesicles, which is a huge, congratulations for that
Thank you.
achievement, and not to be shied away from the Journal of Extracellular Biology. So our favorite ICEV journals. So tell us a little bit about both, and maybe we can talk about the Spirulina one, because that's what we
Sure.
haven't covered yet. Yeah.
Sure. So let's talk about this JEV publication, Journal of Extracellular Vesicles. So this was like I'm trying to say for a long time now. Just it's extracellular vesicles from Helminth against to regulate immune response in host. So uh, we have in this publication, which is titled Helminth Extracellular Vesicles Co-opt Host Monocytes to Drive T Cell Energy, uh, we found that extracellular vesicles from a a worm called a nematode, to be exact, a roundworm, Ascarisum, to be efficient against changing phenotypes of uh, monocytes inside blood, and through that to suppress T cell derived inflammation. And we could utilize them against IBD models in mice, right? So this was a very big opportunity for me. Actually, I sneaked my way into that study. This study was initiated in Aarhus University. So, and I should congratulate and thank Professor Peter Nason and Dr. Ann Barup. So my co-lead, Dr. Ann Barup, it was her PhD project, and this project initiated from Professor Peter Nason's lab at Aarhus University in Denmark. So in Denmark, they did the legwork. So they found as they extracted those extracellular vesicles, they exposed them to PBMCs. So and they found that they exclusively these EVs have been uptaken by monocytes, and that EVs cause those monocytes to change their phenotypes, and those phenotypes are more inflammation repressed, right? They cause less inflammation through deactivating T cell, or suppressing T cell response. What we did, so they were interested in, so in petri dishes, everything can happen, right? Many things can happen that would not happen in animal or human body. So they sent us those EV samples, and we did the the IBD or colitis mouse models. So we routinely we do it here at AITHM at JCU. And what we found that in specific IBD mouse models, those EVs can significantly mitigate the symptoms of IBD. And we found that yes, that would be a candidate for further investigation. Those helminth EV to probably to be used or to be inspired by. We can develop new therapeutics in future, hopefully. So that about that. I hope that I convey it right. But about the Spirulina project. So I love Spirulina. I fell in love with Spirulina probably 3 years ago, and it still fascinates me. First, let's discuss that what Spirulina is. Spirulina is a cyanobacterium. And cyanobacterium means that those bacteria that do photosynthesis, and mostly they are green. They call it blue-green algae as well. So and they are single cells, but they kind of, stick to each other back to back, and probably thousands or even some at some point tens of thousands cells stick back together, and they make a kind of spiral form of spirulina, and they move along in fresh water, in lakes, and they live there, and for thousands of years. Many people from ancient, tribes in various civilizations utilize and then consume this spirulina, either for themselves or for their livestock or pets. So and they made spices out of it. So spirulina is edible. It's not toxic. We have many blue-green algae that are toxic, so they have specific toxins, very fatal toxics actually. But spirulina doesn't have that characteristic, and it has it's a lot of nutritional benefits in spirulina because it has a lot of it produces a lot of proteins, minerals, various nutrients, and so that's not only a food, it's a functional food, meaning that it can have some benefits against some chronic situation like type 2 diabetes. It's now official that spirulina can actually prevent type 2 diabetes and to somehow mitigate it. Obesity, and as well as some type of cancer because it has a lot of vitamins, and also I forgot the name at the moment, but anyway, a lot of benefits. But what we did is actually discover another side of spirulina. So spirulina, like any other life forms, can produce extracellular vesicles. Although it was kind of expected that we find this, but it wasn't reported anywhere. So I seized the moment, I seized the opportunity that, sometimes something is not investigated and you can focus on that. So I thought, hey, nobody worked on spirulina extracellular vesicles. Let's do it. And that's one side of novelty in that project. So extracellular vesicles from spirulina, we found them. Not only we found them, we actually characterized their proteins of extracellular vesicles, and we found 54 unique proteins inside it. It can be divided into 7 superfamilies, 7 protein superfamilies. And we didn't stop there because we are a medical and preclinical research institute. We wanted to see what happens when you inject an EV from spirulina into mice, right? Because it's non-toxic, it's edible. There's a good reason that we don't see actually any effect. So what we did inject them. We did inject them for long term, short term. And what fascinated us was that every time we injected that Spirulina EVs from different batches, various repetitions, we found that there is a lot of inflammation happening, a lot of inflammation like neutrophils coming to the injection site and also M1 macrophages coming to the place and also some sort of T cells. But we don't see any adverse effect in there. So we didn't see any adverse effect, but we see a lot of neutrophils, M1 macrophages, and some sort of T cells. What we found there triggered us. So no, it's pro-inflammatory, meaning that it can induce inflammation, but it didn't cause any, at least visible adverse effect in mice. So what we thought about, hey, maybe we can have a use for that Spirulina EVs. Maybe we can find you can use it as an adjuvant for vaccine. You know what adjuvant does in vaccines? Not especially for subunit vaccines, none of them would work on their own if you inject them, solely. But you have to inject something that actually can that so that the body can pay attention to it, right? So it means that the adjuvants are those chemicals normally that we inject them so that the body can actually trigger, and they're hypersensitive against those and then produce a lot of cells into there. So when the cells come to the injection site, they also come across the vaccine, and they can induce more response against that vaccine because of that adjuvant that is there. So we thought that maybe we could use it as adjuvants, and then we did, and we injected that extracellular vesicles along with some experimental subunit vaccines that we have, and it actually produced at some points 100 to 500-fold more IgG response against the vaccine compared to injected alone. So we found yes, and that was the reason that Spirulina EV can actually be a good vehicle for vaccine industry. We know that so far EVs haven't been approved for injectable medicines, right? But there's a good chance that they will be in near future.
I think so. Yeah, I agree. I think we're on that track. Certainly, it's going to be just the first group or first person or first company to break that ground, and then I think the floodgates will open, and all of a sudden everyone will be using EVs in some kind of therapeutic vaccine or otherwise.
Exactly.
I find that really fascinating. And also, I mean, given that they are edible and non-toxic and whatnot, are there any thoughts about trying what happens if you feed the mice in like EVs from Spirulina? Like, would that also be a potential, seeing as you've already done some research in the IBD area, perhaps they may also have some kind of purpose for gastrointestinal disorders of that kind? Because I love food-derived EVs. I think it's a really understudied area, and Spirulina seems to fit the bill in that case.
Yeah, I mean, definitely. But people have been giving Spirulina to laboratory animals for various experiments nowadays.
Yeah, whole Spirulina, right?
The whole
like
Spirulina, exactly.
Yes.
Exactly, and actually, it affects IBD as well. So it actually mitigates IBD. It found that
Yeah, amazing.
Spirulina shell or Spirulina membrane. So cyanobacterium are like membrane-wise, they're just like gram-negative bacteria. So they have inner membrane and outer membrane, and
So they're
So
really producing these like membrane vesicles, like outer membrane vesicles or membrane
exactly,
vesicles because they're bacteria rather than
yeah,
extracellular vesicles. Yeah.
yeah, exactly. That membrane that produce EVs, so the membrane produces EVs. They might have what you found a safe pro-inflammatory response. But inside spirulina, it's actually spirulina produce a lot of compounds that suppress inflammation. That's very
Hmm.
fascinating.
Ah.
So so
So
that's
maybe the
yeah
EVs are better suited for vaccine and the actual spirulina better suited
yeah.
for. This is what
It's
I
all
wonder. I mean, it's the
yeah
package sometimes, not just
exactly
the
yeah.
EV.
Yeah, exactly.
Ah.
I was thinking about, some thought experiments about, you actually feed them or actually, you know, my thinking was for me, my future or anyone, they're welcome to do that. It's not a idea that I want to keep it for myself. Uh, for topical use, spirulina AV for topical use. So, we have some wounds that, needs to be healed as soon as possible, unskinned, and there is a lot of application for that. We never get to test that, and probably we won't be able to because the funding is drying. It's always in science, some fundings are drying out, and you can't continue on what you're doing. So, but I hope that I could continue on that and to make some other uses for that. However, yeah, topical use is another option, or actually nasal sprays as well for some cases like some tuberculosis vaccines. So we're trying to do that, but with a group that here that we are hoping to do that in the future, that with a group that is working exclusively on tuberculosis vaccines. So I don't know. There's a lot of application. What my thinking was, and again, anybody is welcome to do that, is to use that vesicles as a vehicle for expressing, for delivering recombinant vaccines. I did apply for a grant for NHMRC ideas grant within Australia. So actually having that idea to uh, express and uh, vaccine antigen again on one of the proteins on the cell wall on spirulina, so that it can end up going to the vesicles as well. So that protein can would be a antigen against a hypothetical disease, and that giving the fact that spirulina is uh, you know has pro-inflammatory response and can act as an adjuvant, and you actually added up a layer of vaccine into it, it can be a self-adjuvanting vaccine. So obviously it didn't get funded. So uh,
I
like
know the
most of them,
ideas grant system, and
it's
believe
very
me, it's probably nothing to do with you. And
no, no, no. I mean,
I attended a workshop recently on grant writing, and all these things are discussed, like, is the reviewer in the right frame of mind? Who, like, did something bad happen to them that morning? Are they tired? Are they cranky? Are they like? I feel like
I
you could
know.
flip a coin for these grant outcomes this time.
Yeah,
And
true.
the funding system in Australia is broken, just like the rest of the world, I feel, and the success rates are low, but you just have to
Yeah.
keep trying, keep
What
going.
what what you used to say back in time in my veterinary practice that at some points, when when you don't have a lot of cases to cure and you don't have a lot of visits for your veterinary clinic, they say that the table is small, and the Australia table is small for research grant. We don't have a lot of fundings. That's the problem. And what we allocate when the government allocate for the grants, it's not enough for all the researchers or all the ideas. So what happens in there? They have to regulate it so much that you screen it so tight that only a few can pass this screen, you know. And that's
Yeah.
that's that's the reality. I mean, the United States is obviously I haven't been there, but obviously it's way better. Hopefully with Trump it doesn't stop.
Well, Fortunately, I've arrived just before the current president won, and the NIH was back on its feet again. And now
I
maybe the
know.
NIH will not be
I know.
very happy. We'll see what happens. But yes, it is what it is. This is research, unfortunately. So what I mean, so I'm going to backtrack a little bit. So we actually met at ISEV in Melbourne. You were so kind. You came up and you introduced yourself, and it was very nice to meet you. And that's why, like, when your papers came out, I was like, hey, I met Farouq, and like I was really excited for you. And I guess like I want to know like what's been your experience overall in EV research in Australia? Where are you headed next? And then after that, can we go back and I want to hear about what got you interested in science in the first place?
Well, thank you. You're the kind one, actually, but thank you so much for saying that. So I I honestly don't know research uh, in EVs. To be completely honest, research in general is what I love. Not it's not a cliche when I say that because I always wanted to be a scientist, always,
Well,
from as
that's
far as
good.
I remember. Uh, and I am right now, and I'm grateful for that. But it's not without challenges. It has a lot of challenges, and especially for stability of your career. So at some points, you land on a grant, you'll use that, you flourish. At some points, you can never find a grant, and you're going to change your focus of your research, and then so what I'm thinking about, I fell in love with EV research. It's very huge. There is a lot of potential in it. So many unknowns. You can be really, especially the therapeutic side of it, right? It's just starting, and it's starting very, very robustly, I guess. Uh, I would love to stay, and I'm actively writing grants at this very moment to stay in research. But it all depends, it all depends. I once shifted my career from toxoplasma or soil parasitology to extracellular vesicles, but it might happen, when funding is dried and your career is not stable, you have to change your field. I mean, there's nothing to shy about, and you gotta move on, right? But let's say everything goes on plan, and I could land on grant. I think there is a good chance for me to stay in the therapeutic and biotechnology um, aspect of EV research, especially in drug delivery. There's a lot of fantastic things going on in diagnostics, right? It's very amazing. I mean, how people I mean, some things are weren't there. Many things weren't there last year, right? That are present now at this very moment about diagnostics through EVs or circulating EVs in blood. Biotechnology-wise, I can see that it's already been started. Loading, therapeutics on EVs or, synthetic EVs, drug delivery using EVs. So I would love to, I myself, seeing myself, if everything goes well, if grants go well, seeing myself to continue on that side of EV research for therapeutic and drug delivery. Hopefully, it comes to a conclusion, but no one knows. I mean, research is not a sole focus. EVs don't know that they are named EVs, so we actually name them, we categorize them. We categorize everything. Even bears don't know we're calling them bears, right? It's just science. It just makes sense to us
Yes.
humans, and we learn that. And that's just the beauty of human knowledge, that we name everything. But uh, to me, science doesn't stop at one, research field. Uh, Although I think EVs are a very good topic of research, fantastic topic of research. Uh, if things didn't go well about grant fundings, I could probably shift even though slightly my focus of research. But yes, if everything goes well, I would love to stay on therapeutic side of EVs.
Yeah, I think that's, you can only hope, but I think that's the right way, that's the right direction to go in. I mean, it is a very hot area. I think if you can get, and again, I must stress, it is luck in the grant system in Australia, more than anything. If you can get lucky, and I wish you all the best, then that would be
Oh,
the
thanks so
way to
much.
go, I think. So tell me, you said you knew you always wanted to be a scientist. So when were you like 3 years old, 5 years old, 10 years? Like what? Tell me the story.
Sorry, I know it's a cliche, but I am true when I say that. It was, to be quite exact, I remember my dad bought me a book. It was a picture book. It's a picture, probably kind of encyclopedia, very thin, and it has a lot of very good pictures, very good infographic and pictures. I was born in '89, so when I was 6, it probably was '95, something like that. Uh, and I didn't probably read properly. I could read, but not very well. 6 or 7, I guess. And but I was always amazed by the pictures, just infographic. They were about everything, about space, the common knowledge at that time, about space, layers of cosmos, and it was about atoms and nucleus. I vividly recall some of the pictures. It was the human body, some surgeries. It was about chemistry, about physics, about astrophysics. So it has everything in it. Geography, about satellites and computers. I just enjoyed watching those pictures, and then I kept the book for a few years at least. And when I developed the art of reading, and then I read it, and I actually I liked it even more. So that's how I always wanted to explore new things. So the book was talking about Magellan or Magellan. So I don't know how to pronounce it in English. I read the book in Persian. So the great discoverers, the Christoph Christoph Columbus, or what do you
Yeah, Christopher
call it?
Columbus. Yeah,
Christopher Columbus,
he's
yes.
he's famous here in America. Yeah.
Yeah, exactly. It was about those, about Copernicus, and yeah, why not? I can be one of them, right? That was I fell in love with love, and I still love physics, although I'm a life scientist or a biologist in general. But I still love physics. I still follow physics discoveries and many aspects of physics. I love it. My wishful thinking and my long-time dream is to actually bring quantum physics to biology. Although there are people working on that, obviously, so it's a decades-long research focus. But to combine it to biotechnology somehow, I don't know. It's just a dream. That would be fantastic, just for the sake of the game,
I'm
right?
sure it's a dream that will become a reality one day. It's absolutely
Yeah,
crazy how fast things are moving in those areas.
yeah.
But yeah, that sounds like a really nice introduction to what would be your future scientific career.
No, I mean,
I'll
imagine if you can produce electricity from a tree. How
oh,
cool is that?
that would be amazing. I'll share a story. So I have two boys, and the eldest one just turned 7,
Ah,
and
great.
he knows I'm a scientist, and he has been saying for maybe the last 6 months, 12 months, when I say, what do you want to do when you grow up? He's like, I think I want to be a scientist. And I was like, oh,
Ah.
that's really nice. Like, he knows I'm a scientist. I don't really think he knows what I do, but it's nice. And then I kind of probed him a little bit more recently, and I said, so So why do you want to be a scientist? Tell me, like, what is it? What do you want to do? Experiments? Do you want? What do you want to? And he says, no, I just want to blow things up. I just want to make things explode.
Oh,
And I said, okay, maybe time to think about
maybe.
a different career choice. So not quite the same
Oh my goodness.
thing. But
Yeah.
everyone's
It
different.
probably, I mean,
Um,
in those cartoons, they see that the scientists mix two things and he blow up, and they
Yeah,
get
that exactly. They
crazy.
think, oh, that was fun. No. So I think like this is a nice place to kind of finish up. And I just wanted to ask you one last question, which I ask everyone who comes on the podcast, which is, what do you think will be the next big thing in the EV field? And maybe we've already discussed it if you're going down the drug development kind of pathway, but is that what you think, or is there something else that you think will be the next big thing in the EV field?
Ah, well, actually, I thought about that previously about what would happen with EVs. I think that's my thinking and also wishful thinking that people come around with uh, EVs that approved, obviously, FDA approved, or uh, uh, that can actually can be used to cure some congenital problem through drug delivery or through gene editing technology and
Yeah,
delivering
like
gene
maybe
technology.
CRISPR-Cas9
CRISPR to
system.
some
Mm.
cure some congenital disorders. You and I are parents, and we know that how children are dears to the parents. And at some points, not everybody is lucky enough to have, healthy children. So I think the next big thing, the next, meaningful, interesting thing would be something like this. And if we have a lot of means or tools to do that theoretically, but if you can someone can to uh, delivery side of EVs, I think would be the next big things if something like this happened. Yeah.
Yes, and that's funny you say that because my PhD supervisor, Professor Murray Mitchell, shout out Murray if you're listening, although I don't think you will, he had this theory for many years that one day EVs would eventually be delivered to babies in utero to fix congenital abnormalities or defects or something like that. So that was his very futuristic vision for what
Oh,
EVs could possibly be used for. So, I think that says a lot. So perhaps you're onto the right thing. Um, so, so anyway,
hopefully.
Farouk, thank you so much for coming on the podcast. I really
Oh, thanks
appreciate
for
your
having me. It was
time.
great. Thank you. Thanks for
And
having me.
who knows, maybe I will see you at a future ISEV. Not this one, unfortunately. I won't be attending,
Me
but
neither. Me
maybe,
neither,
yeah.
but but maybe in the far future, probably. Yeah,
Yeah.
I wish you well. Thank you for having me. It was fantastic. I didn't actually expect it like this, but it was greater than I was expected.
Yeah, great. Oh, that's
I'm not
wonderful.
sure.
Okay. Well, yeah, take care and we'll say goodbye to our listeners. Bye, everyone.
Bye. Thank you.